Hello and welcome to a third week lecture of our NPTEL course Introduction to Mechanobiology. In the last week, we had discussed about how nonlinear elasticity can be encoded in a single protein, and in that regard we were discussing about this ECM protein called fibronectin. Okay. So, fibronectin has 3 modules FN1, 2, 3. So, it is a dimeric protein, homodimer, and each of these modules 1, 2, 3 independently fold. Okay. So, within this folding structure we wanted to ask that how are individual domains within this FN3 module respond to forces, and does it correlate with our signature, which is here. If you do thermal denaturation, what you know is the third domain of FN3 is among the most stable, and it takes nearly 120 degrees Celsius for it to denature, in comparison to the 11th domain of FN3, which denatures at 48 degrees Celsius. So, what you have is a vast range of temperature sensitivity, and the question we wish to ask is, does the folding unfolding does the folding dynamics under forces behave similarly to that under temperature. Okay. And in that regard we had been discussing about AFM, and we had shown how what are the different stages in which if you do protein unfolding using an AFM, what kind of a signature do you have which is indicative of a protein unfolding event. So, in that regard, so just to briefly recap, you have a tip, you have a protein, and when you pull on it, so when you indent, you form non-specific bonds of addition with the protein at some random point, and when you try to go up, so you would see in the signature you would have these folds like this. Okay. So, this is my Z position and this is my deflection. Okay. So, the question is, how do we understand how the folding dynamics or the stability of individual domains within this module. So, that brought us to the question that if you have a domain A, what protein construct would you use? Would you use just A or AA or so on and so forth? And what I reasoned was, if you do not know how this particular domain unfolds, then it would be wiser to use a combination like AB, which is repeated n times. Okay. So, A is the domain whose unfolding signature you wish to determine, and B is a known domain of some other protein whose protein signature you already know. Okay. So, what we wish to know is, when you design when you engineer your protein, should you have a protein construct which has this structure versus between these two protein constructs, which one would you prefer? So, let us assume you have chosen this particular construct, which is A repeated 4 times and followed by B repeated 4 times. So, in this case, and let us say, okay, you have done the experiment and this is the output that you get. Okay. This is the output you got once. Okay. So, you see, compared to other curves. So, let me draw another case. Okay. Compared to this case y, for case x, you got this long gap extension in between with almost 0 forces and nothing else, and you have these 2 peaks. Now, you know that these 2 peaks correspond to detachment events. So, they do not really come as a consequence of your domain, suggesting that this long stretch in between is the signature which is representative of your protein construct. But you do not know whether this is really coming from A, because B has not contributed. So, B does indeed does unfold, which means that you should get some unfolding signature. So, in one case X, so if you pick up the protein, such that all these B domains are here and all these A domains are here, B will not contribute to your signature, suggesting that this long gap of almost 0 force is a consequence of your domain A, but you do not know for sure, because you have not gotten any contribution from B. However, let us say you have chosen this alternate design, and you know that B unfolds under force. So, let me draw various representative cases of what you might see. This is one situation. This is another situation. So, what you see here compared to the previous case, where you did not see any other signature from B, you see some signature of B appearing. Let us assume for the time being these 4 are the corresponding of B. So, you still have 2 cases. So, because wherever your tip touches the protein, you are bound to have AB sequences in between. So, which means that if you get a peak from B, then a peak from A you must have. So, you are guaranteed that you will have signature coming from both A and B. So, this is the advantage of having a repeating unit of A, B, and then you repeat the structure. So, this is why people prefer A B whole n as opposed to A n B n, because since you do not have control over where the tip touches the protein, wherever your tip touches the protein, if you have a sequence like A B whole n, whatever point the tip pulls the protein, you are bound to have both the domains A and B pulled up. So, as a consequence you will get the signature. So, this would suggest that both these cases are 2 possibilities in which A has somehow given your signal. So, let us first try to understand what would have happened in this particular case. So, let me redraw this case. So, what you find, disregarding this and the last force, you have a protein which is A B whole 4th. So, you have these 4 forces, force peaks, which are comparable to folding signature of B reported in literature. And you have this long gap. So, for each of these forces, what I can plot is the histogram corresponding to the unfolding force F of u and L of u. So, for these peaks, let us say I get a certain force peak and a certain length peak. So, this is So, each of these lengths L, 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 L will fall in this histogram, and you will have one LU corresponding to unfolded length of B. Okay. So, each of these L corresponds to unfolding of one domain of B, okay, which suggests, so the absence of any force for this peak. So, this is almost 0 forces and a long thing. What is it possibly? I would argue that this kind of a force signature would suggest that this particular domain A is unstructured. Okay. So, let us think of a possibility. Okay. So, what I am saying is A is an unstructured domain. And why, on what basis do I say it? For the simple reason that if you have, let us assume, if I draw a protein like okay. So, these are folded and these are okay. So, now let us assume that this length is L. Okay. Now, because it is unstructured, it will not exactly be present like this. The protein will not have a configuration exactly like this, but it will have a configuration probably in other words, it will still take little bit of a almost negligible force to straighten this particular domain. Okay. You would still require some small amount of force to straighten the unstructured domains. Okay. So, in that case, what this length corresponds to? So, this length corresponds to 4 times length of unstructured domain. Okay. So, this long gap of almost 0 force is nothing but 4 times the length of one unstruck domain of A. Okay. So, this is one possible case where you have a domain A which does not have a structure per se. Now, let us take another case. So, once again these are my detachment peaks. So, these are my detachment peaks. Okay. I know that these 4 correspond to B. Okay. So, folding unfolding of domain B. Now, you have a small amount of length, again 0 force followed by these small peaks. So, now you have 3 species, right. One, the unfolding signature of which you know, and then there are 2 additional things. Okay. So, once again what you can do is you can plot the force peak. Okay. I am not plotting the one for B, but what you have is for this you can get a small force peak, okay, and a corresponding length peak. Okay. So, this is the force, this is the length. Okay. So, let me also draw it for B. Okay. This is for B, okay. And you have 2 zones, okay. One zone of 0 force followed by these 4 peaks. So, this I can assign this force peak and length peak to B, A partly, but you still have this small zone where there is force is 0 and the domain is getting stretched. So, what this suggests is this particular A is partly unstructured plus partly folded. Okay. So, it suggests that you have two zones of A, maybe one part of which does fold and that requires a smaller force to unfold, and another part of it which is unstructured. Okay. This might be another possibility. For the case, okay, for the case let us say A is known to be stable, okay, and you have B which is of course stable. In this case, I might have a signature okay. So, the way I have drawn it, again once again I can disregard the last two forces. So, I have drawn 2 of B let us say and 3 of A. This is one representative curve. So, please note that these protein pooling experiments have to be done several times, so as to generate enough statistics, and that is how you will get the Gaussian distribution. Okay. So, in this particular case I have drawn these 3 A's and 2 B's. Okay. Then I know for sure, if I know the protein folding signature of B, I know what is the folding signature of A, and I can confirm it by knowing. So, you know that if you have various residues, okay, so you know that the total unfolded length equal to number of residues into 0.38 nanometer per residue. Okay. This is the rough length of one amino acid. So, you know the unfolded length, and this formula can also be used for the case where A is unstructured plus partially folded. Then you can find out, know for sure whether in this case your protein A forms these two parts, because you can add up this length and this length. Okay. So, this case you know that for these two put together you must get the overall unfolded length. Okay. So, for the case I have drawn here, in this case I have shown 4 peaks, small peaks and one unstructured length, right. So, you know that 4 times unstructured length plus partially folded length into 0.38 nanometers should give you the entire length. Okay. Now, let us think of one possibility. Let us say in the actual domain, so you had 15 domains of FN3 module. So, you might have neighboring interactions between, let us say FN3 1 and FN3 2. Okay. So, neighboring domains stabilize each other. In this case, what you might see, if you have a protein construct which comprises of versus okay. So, you had one protein construct where this domain was coupled with this B, which is whose unfolding signature you know, versus this case in which 1 and 2 are next to each other, and this domain is repeated n times. So, what you will observe is the force unfolding signature. So, this F1 let us say corresponds to FN3 1 domain here, and this F2 corresponds to FN3 1 domain from here, from this construct. You might see F2 is greater than F1. Okay. So, if there is a stabilization by neighboring domain, you might see the F2 force that you measure to be greater than this F1 force. Okay. Versus if there is no neighboring interaction, then both F1 equal to F2 is what you will get. So, this is no stabilization and F2 greater than F1 in the case of stabilization. Okay. So, using this strategy, you can theoretically determine the unfolding force signature of all the domains 1 dot dot dot. Okay. So, if I were to again write down the strategy, form or engineer different protein constructs verify that they fold three, okay, determine unfolding signature. Okay. So, this way you would be in a position to map the forces for each peak. Now, let us assume, okay. So, you have determined the unfolding signature for all the entire protein. Now, let us say, okay, you have these cells sitting on ECM proteins, okay. Let us say sitting on fibronectin matrix. Okay. This is cell type A, this is cell type B. Okay. So, the unfolding signature tells you that signaling will depend on the magnitude of forces exerted by cell A versus cell B. Okay. The next question is, the rate of pulling. So, in addition to pulling, so one is pulling force, second is pulling rate or rate of contraction. Okay. What would you expect to see some differences between two cell types which exert pulling forces at completely different rates? And the answer to that is indeed. Okay. So, one way of designing that experiment would be, okay, so you have your protein and what you change is the rate of pulling or the tip speed. Okay. So, if you change the tip speed, you can simulate, so faster speed is equivalent to higher pulling rate. So, using this, what has been observed is, if I were okay, this is my force and this is my frequency. Let us say for a given domain, this is at a certain tip speed v. What has been observed is for many proteins and many domains, okay, if you pull the protein at a tip speed v prime which is less than v, okay, then f prime is also less than f. Okay. In other words, if you pull a protein slowly, it takes less amount of forces to unfold the same protein. Okay. This is something very interesting, but it also says that even if the same magnitude of force is exerted, the rate at which a protein is pulled also matters. Okay. With that, I stop here today. In the next class, we will continue from here, discuss little bit more about one more aspect about protein unfolding, and then we will go on to study focal adhesions. Thank you for your attention.